evening. Welcome at the second biological Thursday this semester. I see many faces of people from abroad, so maybe I would utilize this opportunity to stress that Biological Thursdays represent the longest series of seminars, at least devoted to biology, but probably devoted to any natural science in the Czech Republic. It started already before the revolution in 1989, and we have these seminars like every Thursday in both semesters. Some of them are in English, as you will see. Still, the majority is in Czech, so for many of you it's not relevant, but just I just felt that it is important to point out that this is really considered to be a major series of seminars for wider public. Before I introduce today's speaker, I would just point out that the next seminar or next talk will be talk of René Kysely and Luboš Peške. And it will be in Czech, but they will be speaking about a very interesting and new topic about the domestication of horses, because maybe some of you have realized that there are really new findings concerning the whole history of this domestication. So those who speak Czech, please come. And now it's time to introduce today's speaker. Brian McGill is, for me, probably the most interesting and creative ecologist in the world. Of course, I'm biased because I do similar stuff to his stuff, because he's doing macroecology in very broad sense. He was dealing with patterns in abundances, diversity. He often goes kind of deeply to conceptual issues concerning ecology in general. And in last, let's say, decade or so, he's interested also in biodiversity, in the Anthropocene, basically current changes of biodiversity, and again going deep into the conceptual issues concerning what we even know about biodiversity, how can we measure it, and even how can we communicate about it. And this will be the topic of his talk. Please, Brian.
Oh, thank you, David. I think I've got to be in Prague twice this year for science, so that's good. I'm enjoying it. Yeah, so I'm going to talk about biodiversity broadly. It's really 4 or 5 talks that are all going to center around the theme of biodiversity, but one of them is going to be more social sciencey, some of them are going to be very quantitative, some of them are going to be very empirical. So I'm going to start with a really obvious question: what is biodiversity? And the term emerged in the late 1980s. Doing some research, it really was, I think, almost accidental. Everybody says E.O. Wilson introduced biodiversity in 1988, which is not technically true. You can find it in the literature before then. And additionally, he was just running a seminar program on biological diversity and just made the word biodiversity to fit in posters very well. And so, but you read the book, he doesn't have this big conversation about biodiversity as a new word or a new concept. But certainly by 1993, but nonetheless, I think it's important to note, all of these were policy contexts. These were people who worked for the US government, or these were National Academy seminars trying to get the government to pay attention to an issue. So it very definitely emerged in a policy context. It was very firmly in that context already by 1993, so 5 years, quick movement in a short amount of time. They defined biological diversity as meaning the variability among living organisms from all sources, including, among others, terrestrial, marine, and aquatic systems, and the ecological complexes, excuse me, of which they are part. This includes diversity within species, between species, and of ecosystems. That's a pretty broad definition. It's pretty hard to argue with it, right? But, and it's good for a policy treaty that you want to be inclusive and include all aspects of biodiversity. It avoids the mistake made in the United States with the Endangered Species Act, right, where we just focused on the species level. And for sure, the word biodiversity is taking off. Things like conservation and wildlife had their peaks in the 80s and 90s. Biodiversity, this is a Google Ngram. It's maybe flatlining, but it's got more of an upward trend than some of the older terms. It does seem to be the term people have centered and homed in on lately. Okay, but as a scientist, we don't need this gushy policy word. We need something that we can measure, right? This is referred to as making it operational. And the notion was first introduced in physics. Probably doesn't surprise you, right? In physics, if you can't measure it, what good is it? Rob Peters, an ecologist, very strongly made the same notion that if it's not an operational concept that I can measure, it's a problem. And really, biodiversity at the moment is the opposite of operational. So we would like to, move towards a place where we can operationalize biodiversity. It's a unique, defined set of operations to measure something where different people can come along, repeat those same operations, and get the same answer. So I'm just going to paint a picture for a minute. I'm not going to show you equations or anything. But the breeding bird survey covers a route that's about 40 kilometers long. It makes a stop every half mile and records every bird that you see or hear. And these are just the warblers, so a subset. This is a route near my house back in Maine in the United States. And if you look at this, there's 17 species of warblers that appear. They're not, not the most glamorous birds, but they're pretty nice birds. Okay, so that might sound like the diversity of warblers is 17. Nice, simple, solved problem. But, and to point out, we do have, these are well-defined taxonomic units. They have names. This is just to point out that even taking the color out of the picture, I would suggest take some of the biodiversity out of the picture, and we have to stop and think about that, right? Because there's still 17 distinct pictures here, but taking the color out does have some effect. And then it gets more complicated. In 2006, we only had, 15? This one was missing, this one was missing. If you come back in another year, it's a different story. This one's back, but this one's gone, this one's gone, this one's gone, right? And so we could do a count. How many years is each of these species of warbler present across a 10-year period? And it varies from always to just barely, right? We could also look at abundance because these birds are not at all equal in terms of number of individuals. Some species are quite common and some species are quite rare. And this is, to plot the full data, this is the full story of what's happening. Each one of these lines is a different species, trends through time. This is a log scale. And we have a number of species that come down here to go extinct and flicker in and out, right? So that's the data we have to work with. What's the biodiversity of this system, right? That's basically the question I'm trying to answer or pose. Just keeping it simple with species richness, the average species richness is this. It's 11.3 or something, which is actually not a number that's ever observed, right? We never see 11.3 species. The cumulative species richness through time gets up to 17, like I showed you, but the observed species richness at any point in time fluctuates. And maybe we need to take abundance into account and the fact that really if you go back to the abundance, there's really only about 3, 4, 5, 6 species, right? The others are so rare, maybe you don't count them or they don't count very much. Whoops, so Shannon diversity is much, much lower, right? So I'm still groping around this question of what is biodiversity and how could you take something that's complicated and measure it? This is just to show you that I was talking about challenges through time, but this is challenges through space. This is Barrow, Colorado Island, so trees in Panama. This is just 4 species out of 220 something species in this plot, but they're not spatially equally spread out through space. And so depending on what size transect we put down, we're gonna get very different stories. Okay. So, and just to acknowledge, I'm talking about taxonomic diversity, but of course there are lots of people for good reasons who are interested in functional diversity, phenotype, phenology, other things, phylogenetic diversity, genetic diversity, And in fact, if you go back to the CBD, the Convention on Biodiversity, all these things are incorporated in there. Just one quick slide on trait diversity. This is a former graduate student, Julie Messier. This is, I think it's 120 species of trees in Panama, same plot that I just showed you. And this is one functional trait, leaf mass per unit area. And you can see there's enormous variability across species there too. All right, so I'm going to argue that to really talk about biodiversity, we have to get serious about scale. And I'm going to suggest that actually the answer cannot be given without talking about scale. So biodiversity is problematic. Most things in science are classified. Physicists came up with this, but this extends into most of chemistry and biology. They can be extensive. So if I double the area, I have double the number of measurement. So number of molecules in a gas is the classic example of an extensive variable in physics. Or they can be intensive, where if I put two units together, I just average the original measurement. So temperature is a classic example of that, right? If I have two separate vats and I mix them together, I get temperature, and it's intensive. And so extensive increases with scale linearly, and in fact, a one-to-one line. I guess it's not one-to-one, but it's linear. And intensive variables are flat. They don't change with scale. You're just averaging across bigger and bigger areas. And of course, as many of you probably know, this is actually what biodiversity does. It is not intensive, it is not extensive, it's nonlinear. This is where most of the interesting behaviors of biodiversity come from, and it's where most of the problems of biodiversity come from. So I want to ask a simple question. These are the kinds of questions policymakers want answered all the time. There's an invasive species. In this case, it's a plot from John Chase. It's a honeysuckle. It's an invasive bush, nice pretty flowers. Humans plant it all the time, but it's an invasive species. Did it increase or decrease biodiversity? This seems like a binary question that should be easy to answer. But to answer it, we do have to look at scale. And the first question is, should we scale by sample size, number of individuals, or should we scale by area? And it turns out, unfortunately, the answer depends on which one we do. So this is scaling by area. Here's the invaded plots and the uninvaded plots. Here's species richness at the local scale. Single small quadrat plots. Richness is clearly lower. It's statistically significant. If we go to a larger scale, richness is a little lower, but it's not statistically significant under Monte Carlo reshuffling. But if we scale by sample size, number of individuals, actually we get the opposite answer. The invaded plot has a slightly higher species richness. It's not statistically significant at small scales, but at a large scale, it actually is statistically significantly more species. And these are different measures of richness. This is a richness just looking at species richness versus area. And this is rarefied richness where we're controlling for sample size. We get the opposite answer. An invaded plot has more species. So going back to this notion of operationalizing things, making things measurable, this is kind of problematic, right? We actually get the answer to flip depending on how we scale things, and which scale, also depending on which scale we look at. And it turns out in this particular case, one of the big problems is the real impact of the invasion is not on species richness, it's on the number of individuals. These are bushes coming into a herbaceous landscape. The bushes displace lots of individuals, and so they don't necessarily have a huge impact on species richness, but they have a massive impact. A quadrat goes from about 75 individuals, 87 individuals, to about 10 individuals. So that's actually the biggest impact is on N, not on S. So I'm going to argue that rarefaction curves, which have been around since the 1960s, is actually one of the best tools we have for trying to compare biodiversity and answer questions like, does this invasion increase or decrease species richness? And it turns out these are the rarefaction curves, the average rarefaction curve for an invaded and an uninvaded plot. And you can see that so species richness is on the y-axis. The difference in species richness is incredibly small. It's there. The uninvaded plot is always higher than the invaded plot, But the big change is this, the invaded curve stops right here at 7 individuals, and the uninvaded curve keeps going on, and that's where you're picking up the extra species. So the reason I like rarefaction curves, number one is they include scale, right? So they're letting you do comparisons of multiple scales at once. If these curves crossed, you would actually get a flipping. I think actually maybe they do cross down at the bottom where they look like they're on top of each other. You get a flipping. And so you can actually do a comparison of richness by scale, just by which curve is higher. But they actually contain quite a bit more information, right? So species richness, which is hill number 0, for those of you who do biodiversity indices, are at the height of the 2 tips of these curves. Rarefied species richness, which is where you control for the number of individuals, is just by taking a vertical line, so you can compare things. And it turns out, actually, it's been proven that the pi evenness, Simpson evenness, is the slope of the curve between n equals 1 and n equals 2. So you have the hill number 2 down here, and you have the hill number 0 up here. That kind of implies that the hill number 1 is somewhere in between. That's not mathematically precise, but it is actually, in a certain intuitive sense, true. So you're getting the whole spectrum of different metrics of diversity in terms of the amount of abundance, right? Relative abundances. If you have many rare species, that still shows up equally in raw richness, but it does not show up equally in hill number 2 or pi richness. So it's taking relative abundance into account as well. And this graph shows, this is the same, actually this is a different graph. This is for a made up community. Whoops, sorry. Community with 7 species, 1, 1, 1, 2, 5, 10, 50 individuals, gives you this rarefaction curve right here. This curve is the slope of the rarefaction curve, so it's basically the derivative. It's a discrete derivative. And this curve shows how much of the derivative is contributed by each of the separate species, right? So you can see for n very small, most of the contribution is from the really common species, these species with 10 and 50 individuals. And over here, for n really large, most of the contribution is for the rare species. So what's causing the curve to go up initially is the common species, and what's causing the curve to keep going up is the rare species. If you just renormalize this, so it sums to 1, you can see quite clearly that it's the common species at the low end, and it's the rare species at the high end. So the rarefaction curve is actually packing quite a bit of information. It's not just allowing comparisons, it's actually packing the whole notion of abundance and diversity indices and evenness indices into these rarefaction curves at the same time. Okay, so if you imagine species richness as just drawing marbles, colored marbles from a jar, which I confess seems like a big imagination, but just go with me for a minute. You know, reaching in, the species richness is going to depend on how many individuals you take out. It's going to depend on the total number of different colors of marbles in the jar. It's going to depend on if 90% of the marbles are yellow and 10% are all different colors, you're going to get lots of yellow, so richness is going to go down. And it's also going to depend on this pattern I showed you, the spatial structure, right? If all the yellow marbles are at the bottom, you're actually not going to sample yellow marbles even if they're the most common, right? So the 3 things that are driving species richness are the number of individuals in the sample, the spatial clumping of individuals, right? The spatial structure. This red species is almost all in this corner. This cyan species is all in this corner. And also the relative proportions of rare versus common species. So those 3 things all come together in a rarefaction curve. And with John Chase and a number of other people, Dan McGill and bunch of other people, we've developed an R package that can actually use rarefaction curves. And by doing different kinds of randomization and null models, you can separate out those 3 components of species richness due to abundance alone, which is what we saw in that invasive species case. The change in species richness due to, right, so we have right here, change in species richness due to number of individuals is strongly impactful, but it's in a negative direction on species richness. The proportion of rare species is plotted here, and the effect of spatial aggregation is plotted here. So you can actually break the three components that drive species richness at a local scale into three separate pieces and calculate their weights. Yeah, there we go. So there's an R toolkit, it's called MobR. It produces these graphs for you, so it's hopefully pretty simple to pick up and use if you're interested in that. Okay, so maybe I convinced you, hopefully I convinced you rarefaction curves are a great way to look at the kind of high dimensional complexity of biodiversity, right? Started out with the warblers, they're a high dimensional complexity. Rarefaction curves, it's a graph, you have to look at it with an eyeball, but you can do statistical tests, as I just showed you in the last figure. Rarefaction curves are a very useful way to capture the complexity of biodiversity. Problem is rarefaction curves are extremely flexible, and now I mean this in a literal physical sense. So if we take a community where every species is equal, we get a linear rarefaction curve. If we take a species where the species are all packed in clumps, so you encounter all of one species, all of the next species, all of the next species, you actually get a concave down, convex up curve. And the more typical species abundance distributions, you get something in between, right? So you can have enormous variation in the shape of a rarefaction curve. And in fact, people tend to think about these curves. We have this intuition that comes out of something like calculus, where we think of a power law, or we think a log curve, or something that has this increasing, decelerating shape, and we don't think about these curves crossing. But this is just a sample data set of marine invertebrates, benthos invertebrates. And we had 120 pairs of pairwise combinations of species, and 53 of those curves crossed each other. And this is another version of the same thing. Actually, I think this is for trees instead of the marine benthos. But out of about 200 pairwise combinations, 100 didn't have any crossings. But a good 80 of them crossed one time, 10 of them crossed twice, and a non-trivial fraction, I think is 5 or 6 of these curves crossed each other 3 times, right? Which you don't think about these smooth curves crossing each other. But the point about that is when these curves cross, you have a scale break, right? Species richness is higher on one curve for, at high scales, and species richness is lower at smaller scales. So you actually have a flip in sign of the impact of whatever treatment, invasion, fertilization, whatever treatment you're looking at. Anytime these curves cross, your answer actually changes direction from an increase to a decrease, or from a decrease to an increase, depending on which scale you measure it. And that's kind of disturbing to me as a scientist. You would like to think there's one answer, but actually scale is just really important in ecology, and there's no escaping scale. So this is a meta-analysis we did with a bunch of different studies. And this is the effect size. 0 is no change, so it's a log ratio. Effect size at the smallest scale and at the larger scale. So we just took studies that looked at species richness change at 2 scales. And as you might hope, most of them are in this quadrant, which is increase, increase at the small scale and large scale, or decrease, decrease at the small scale and the large scale. But actually, I don't know what that count is when you include the ones that are really close. It's a non-trivial fraction, where even in these published analyses, this is not a theoretical thing, the sign of the effect on species richness change depending on which scale you measure. And these are not crazy arbitrary scales. These are the 2 scales that experimental people in the system chose to measure, right? And they actually had a change in sign. The other thing you'll notice is because the slope of the line is less than the 1-to-1 line, it means that generally the effect size is decreasing with scale. As you get to bigger scales, the effects on species richness are changing, which is probably not that surprising. So here's kind of the money test. This is a nice figure from a paper by Jack Lennon and others looking at species richness in the UK. And this is a 10 by 10 kilometer pixel. This is a 90 by 90 kilometer pixel. These are 30 and 50 intermediate scales. This ellipse, if you can see it, is roughly London. So the question is London a species rich or a species poor area? It completely depends on scale. If you look at this, actually London looks like a fairly species-rich area at the fine grain, right? But if you look at the coarse grain, it's not the worst, but it's actually a pretty depauperate area, right? So this is not, you know, this can seem theoretical when I talk about curves that are really flexible and bendy. It is theoretical, but this is, right, this is a direct policy relevant piece of information that ecologists would like to communicate. And the answer about species richness and the effect of, you know, one of the biggest cities in Europe on species richness is it depends on scale, right? Over here, these dots are the top 5% richest pixels in Great Britain, and actually a lot of them are inside London. But when you go to this scale, none of them are inside. In fact, they're all in the Lakes District parks, the national parks up here. So scale dependence is critical when you talk about biodiversity. So to summarize this, biodiversity changes can actually change sign depending on scale. So at a minimum, you need a scale explicit approach, something that's scale aware to do these comparisons about does this increase or decrease species richness. Rarefaction curves give you that scale explicitness. They also pack in a lot more information about abundance and changes in factors like total abundance in a community. And they also, with this MOBR package, actually let you break things into, you know, what is driving the change in species richness? Is it a change in the total number of individuals, which is what we observed in that case with the honeysuckle? Or is it a change in the rare species? Or is it a change in the spatial aggregation of the species, the spatial structuring or layout of the species? And I could give you examples of experiments or studies where all three of these are the dominant factor. So when species richness changes, there can be different things happening. And I already plugged the MOBR package. Okay, so I got you to play along by saying just pretend like we're drawing marbles from a jar. Now I'm actually going to get serious about drawing marbles from a jar. We tend to, typically in ecology, have a reductionist view of the world. We start at the local scale and see causality flowing from the local scale to the regional scale to the biogeographic scale. The rest of this figure is not important here. I could give you precise definitions of what I mean by those scales, but I'm going to propose, let's just see how far we can go by saying things go in the other direction, that the local scale is just an echo, or if you will, a sample of the regional scale. Let's just see how far we can go. If we can't go very far, then this hypothesis is correct. If we can go pretty far, we start having to take this hypothesis more seriously. Okay, so we have a regional pool, which is a bunch of marbles in a big area. And we take a sample, and the only parameters here are, well, the number of species, the number of marbles in the region, and the relative abundances, right? How many rare and how many common species there are. And so that defines the regional pool. The local sample, let's literally say, you know, let's say the number of individuals sampled is proportional to area, which is not a bad assumption. And let's just say that we're sampling marbles from a jar. How far does that get us? Well, so the mathematical machinery is you need a sampling function. And this is the probability. So if you take something of a size little a, and you sample from a region of size big A, and there are n sub I, big N sub I, number of individuals of that species in the big region, what's the probability of seeing little n sub I individuals in the small one, right? So this is just sampling. The A and the A are just like getting the areas right. And this is how common the species is regionally. And this is a probability distribution primarily on, did I draw 0 individuals, 1 individual, 2 individuals, 3 individuals, 4 individuals, 5, up to 100 or however many I can go, and put a probability distribution on that, right? And the key point of this is once I define the sampling function, the species area relationship is a simple formula of the sampling function. The local species abundance distribution is a simple function of the sampling distribution. The occupancy abundance relationship is a simple formula of the sampling distribution. Beta diversity is a simple formula of the sampling distribution. So this is a really central notion. The idea has been spread out and developed across a number of different papers. This is a paper that kind of reviews this linkage between all these different things. But the one hand, that's kind of nice. It really boils down many of the patterns of macroecology and ecology and conservation biology into one formula. We just have to get this one formula right. So if we are literally just sampling marbles out of a jar, this should be the binomial distribution, right? Binomial has no correlation between individuals in the sample. And so let's just see how far we can go with the binomial distribution. So here's 3 major patterns in macroecology. Here's the species area relationship. So this is Barro, Colorado Island, trees in Panama. And they have a 50 hectare plot, so we can go from 0 to 50 hectares. We can take each 1 hectare plot and count the number of species that are in that alpha diversity in each of those plots. And of course, it's not the same number in every plot, so we have 50 plots. So we have a histogram of species richness. It turns out the average richness empirically is 91. And this is an occupancy abundance relationship. So this is predicted versus actual occupancy. And you notice that those 3 patterns actually come pretty close just with a simple notion that we're doing a binomial random sample from the regional pool of the half square kilometer and just sampling individuals, right? There are some errors, but actually what's interesting is the errors are systematic, right? We're systematically over-predicting species richness on the species area curve, which also shows up here, right? This is really the case of hectare equals 1. We're systematically over-predicting alpha richness, and we're actually systematically over-predicting occupancy, the number of the 50 hectare plots that are occupied versus what is actually occupied. And so when you think about this, what we're really doing is we're having the individuals too spread out in space, and so that you are getting more individuals into each of those 1 hectare plots, and you're increasing the occupancy of these plots. So the missing ingredient is actually the fact that individuals are clumped in space. Individuals are not placed randomly or uniformly in space. Individuals show clumping. So these are different plots of different, all the individuals of different trees in BCI. Some of them look more uniform, but there's some clear clumping in holes here. There's some very clear clumping in holes here. And this is actually the sampling function. So this is the number of individuals. This is the number of times we found 0 individuals, the number of times we found, I guess I don't have it broken down to the level of 1, but the 0 to 5 individuals, 5 to 10 individuals, 10 to 15, 15 to 20 individuals. And if things were spatially random, we would expect a Poisson distribution, which is this black line. But what we actually observe is this over-dispersed distribution. There are many, many more cases of 0 individuals showing up in a hectare, and many more cases of 40, 50 individuals showing up in a hectare. So there's over-dispersion, and so the binomial, that's the problem with the binomial, is it doesn't have over-dispersion, and the over-dispersion is because of this clumping, right? If this individual's clumped over here, we'll get a bunch, and if it's not over here, we'll get a bunch of zeros. And so there's a nice paper by, I deleted a slide, and that's the slide that are the authors. It's Tommaso Zilio and Feng Liang He, that they invent a new distribution, which is basically, it's like the negative binomial, but it extends into the Poisson realm, and it lets us do the sampling function with clustering. So you have to introduce one more parameter, K, which is the clustering. This is the extreme case where I'm letting K vary for every species for every scale, so it's badly over-parameterized. It should do well, but it does do well, right? The species area curve, biggest deviation is about 5 or 10 species. The predicted species richness has come in much closer, and the predicted occupancies come in almost perfectly. That's too many parameters to be realistic, but we can actually boil it down to 2 parameters. If we assume K is constant across species and K only varies with scale, right? So we just fit a line between K and the number of hectares, right? K and area, so 2 more parameters, just a linear line. We can actually get almost, actually even slightly better predictions, right? So here's the predicted species area relationship. Here's the predicted versus observed species richness per 1 hectare plot. And here's the predicted versus observed occupancies, percentage of hectare plots that are occupied. So the answer is pulling marbles out of a jar comes pretty close to predicting local community plots. Pulling marbles out of a jar, where we actually measure how close marbles of the same color are to each other, and using this clumped or negative binomial distribution actually does extremely well at predicting most of the properties we talk about at a community level for a local community. So this is just, so the point I want to make here is that there's a scale break. So up to about 100 by 100 kilometers. I've been doing trees, I just switched to birds on you. So this is birds of North America. But again, just this notion of sampling individuals randomly with some aggregation does an extremely good job. This, yeah, well, so the red line is MacArthur's species area relationship plotted, right, for a species area relationship. And I don't have it plotted on here, but the simple sampling process that I described does an extremely good job of predicting the species area relationship up to about 100 by 100 kilometers. And then when you get to 100 by 100 kilometers, you may know that the species area relationship shows a scale break and it changes its second derivative. It goes from decelerating to accelerating again, and it goes back up to a slope of 1 to 1 at very large scales. And so the marble sampling that I described to you describes the local communities at that smaller scale. It's really just picking individuals out of the regional pool with some clustering. If I get a yellow-throated warbler, it's more likely that the next individual I sample will also be a yellow-throated warbler. But basically, random sampling explains the pattern up to that scale. At the larger scale, you can do something very similar. There are a series of papers that have done this. If you know Whitaker's, Robert Whitaker loved to do these plots of these bell curves of abundance across a gradient. And if you just extend that into 2 dimensions and you make these 2-dimensional bell curves and you put them down at random, you can actually predict the species area relationship at the larger scale quite well. So both of these really involve the notion of randomly placed individuals, except for the fact that there's a clumping, right? The clumping comes in the sampling here, it comes in the fact that there's a peak in the distribution here. So what does this all mean? I get this question a lot. I basically, in one level, seems like I just said all of ecology is random. I don't want to leave you with that message, so let me try and correct that message. I think what it means to me is we need to be paying much more attention to the larger scale, the regional pool, and maybe, obviously for other reasons, if we're looking at habitat preferences, we should study the local scale. But if we're looking at biodiversity, conservation, community processes, really the signal is at the regional scale and not at the local scale. And that kind of makes sense, right? As sample size gets bigger, error gets smaller, stochasticity decreases. As you get to bigger scales, environmental determinism becomes more important, or at least it becomes easier to measure, right? Measuring microclimate is incredibly hard, whereas anybody can go on a computer and pull down WorldGlim or Chelsea or something and get large-scale environmental variation these days. This is, I think, a key point. Species interactions don't necessarily disappear, but they become less pairwise. It's no longer about this species versus this species, but it's like, yeah, there's a bunch of warblers in the forest and they're all doing about the same thing and I'm competing against other warblers. It's not a pairwise interaction like you get out of Locke Volterra. And the processes driving regional pools are ultimately evolution and speciation. So it pulls those processes in as more important. And dispersal also changes as you change scale, right? At small scales, it's about source sink dynamics, mass effects, these kinds of things. At large scales, it's really about, has the species ever made it into there or not? It's about colonization dynamics, vicariance dynamics. And I would argue it's actually good news that we should focus on the large scale, because that's also the scale at which humans are impacting systems, right? Very rarely is a human impact a 1 meter by 1 meter square quadrat, right? Humans have national parks, and then we have urban areas, and then we have exurban areas, right? So the scale of human impacts is tens of kilometers as well. So I think there's a nice congruence between this notion of studying regional scales and the need for conservation. Okay, global change. David mentioned I'm working on global change. I'll go through some of my older work fairly quickly, and then I've got some newer work that I'd like to present on that. So global change, the Millennium Ecosystem Assessment talks about 5 major types of global change. There's land use change, over-harvesting, invasive species, climate change, and chemical change, which we usually think about as pesticides, harmful chemicals, but actually the biggest chemical change is eutrophication, fertilization, right? I find it kind of interesting that everything humans have done have been a factor of 2. We've taken over about 50% of the land surface. We've removed about 50% of the biomass, which is all trees, right? Because trees are most of the biomass on the land. We've doubled the amount of terrestrial NPP, right? We've doubled the amount of NPP going into the ecosystem every year. We take about 50% of the fresh water on land every year. Oh, there we go. We've doubled the nitrogen input to the biosphere. Very soon we'll double the amount of CO2 in the atmosphere. Just a matter of a decade or so, I think, maybe even less at this point. We've arguably taken more than half the large marine fish out of the ocean. Marine NPP is not quite as simple a story. We've reduced it by about 8% or 25% in coastal areas. So huge impacts that humans have been having. We should be able to find a signal of this. So this is a paper with Anne McGill, Maria Dornalis, and Nick Catelli, which we published in 2015. And this paper, kind of like the first part of my talk, really was, people were saying, well, is biodiversity going up or down? And we basically said that's an ill-formed question. You can't ask or answer that question. Because biodiversity has multiple components. It has temporal beta diversity, change through time in the composition of a community, alpha diversity, beta diversity, right, change across space, and biomass or abundance, and that these answers could be different at different scales, just because, in particular, just because species richness is declining globally does not have to mean species richness is declining locally. There's, I don't know, 9, 10 zeros in the difference in scale between local and global. And so you don't have to have the same processes and the same patterns happening. So we argued there's 15 distinct different kinds of biodiversity. And this was our kind of guess, approximate guess of what's happening to each of those. Some of those turned out to be right, some of those turned out to be wrong. You'll see at the end of this section, I'll show you a graph where we basically have collectively as a group of all scientists filled out this graph. First off, to acknowledge biomass, we've taken 50% of the biomass off of land. So 50% of the large fish biomass out of the sea. So we've taken biomass for sure. There's no, if that's your definition of biodiversity, there's no argument about that. On the other hand, if you look at local species richness, so this is species richness, and these are time series, and each line is a different time series, it's more complicated. We have declines, but we have increases, we have steep increases, and the net trend is close to 0 for local species richness. You guys, this is not a surprising graph when I put it up now. When I first put it up in 2014, I got kind of yelled out of the room. But to me, the more interesting graph was always this one. This is turnover through time in a community. So you can basically, this is a Jacquard index, but it's basically the percentage of species is the same as when you started. So if we're right here, that means after 60 years, only 40% of the species are the same as what were there when I started 60 years ago. Right, so this is a change in composition through time. And what we found is that there's, on average, depends on the taxa you're talking about, but averaging crudely across plankton to trees, we found 10% decline per decade. Right, so after 50 years, you have more than 50% of the species have turned over. And we don't know what the null model for this is, but we tried several null models, and they all suggested this is rapidly accelerated versus natural processes. Right, if you put in neutral. So everybody talked about this graph, but I actually think this graph is the more important graph. We're seeing turnover. We're not seeing, at a local scale, loss per se. We're seeing turnover. Just very quickly, this probably shouldn't come as a surprise given my talk, but the pattern of change in species richness depends on the scale you're talking about. It's pretty close to 0 at local scales. At larger scales, at least in some taxa, species richness has actually increased That's a function of invasive species, right? At least on the mainland, invasive species rarely cause species extinctions. Islands they do, but the mainland they don't. So invasions basically increase regional species richness. And then globally, we are seeing declines in species richness. This is just taking the same data set and looking at individual species. So not species richness, but individual species. This is the slope of population through time. It's very strongly centered on 0. Which, you know, it's not consistent with some other stories you hear about how 70% of vertebrates have disappeared. But I also take a moment and look at that shape and think how you would characterize that shape, because I'm going to come back to that a little bit later. Just to take it back to our warblers, we see the same thing. Some warblers are trending up, some warblers are trending down. Here's a summary graph. Interestingly, these are doing the opposite of what we see both in Europe and North America, where the common species are increasing and the rare species are decreasing. Most of Europe and North America, we see the opposite. Common species are declining and rare species are increasing, which would be good news. But nonetheless, the warblers show diverse responses in trends. Okay, so this is the review paper that I mentioned across lots and lots of different papers. And every one of these numbers is a paper. These graphs are complicated, so let me unpack it. So looking at species richness, we can imagine declines, no change, small increase, increase, right? And we have to look by scale. And this is an attempt to summarize. So at the global scale, richness is declining. At the regional scale, richness is increasing, or weakly increasing. It's somewhere in this range. At the local scale, we're increasing to no change. We're somewhere in this range. If we look at temporal turnover, doesn't matter which scale, humans seem to have greatly accelerated temporal turnover, right? Local, regional, global scales were way up in this end of the spectrum. If we look at spatial beta diversity, this analysis of published papers suggests that it's declining. I'm going to argue that that's not correct, and I'll show you why in the next slide, but I think there's a publication bias in this particular analysis. And in terms of abundance or biomass, at the local scale, it's kind of flat. You can find places where it's gone up or it's gone down, and this kind of makes sense. There's places where we've built national parks, and there's places where we've done restoration, and there's places where we've gone in and paved things over. So it's going up and down locally, but it's hard to find a major trend. Regionally, where you're averaging across the parking lots and the cornfields and the monoculture crops and the national parks, biomass has gone down, and globally, the only one I know of is trees, it's gone down. Okay, so started with that template of the 15 types of biodiversity, and here's what 96 papers have concluded to date. It's a complicated and interesting story. Some cases of no change, some cases of increase, some cases of things not as bad as we thought. I did want to come back to beta diversity. This was a recent paper published with Shane Blows and John Chase and much of that group that I identified before. And we took a bunch of, well, the same set of time series. And so it spans taxa from plankton to trees to birds to mammals. And we calculated whether beta diversity was increasing or decreasing. It's decreasing by a very, very small amount. And again, the real story here is the spread, right? There's a large variability of increase and decrease, right? And this is probably the best graph to look at. Locally, some places are showing decreases in beta diversity, which would also be called homogenization. Other places, they're showing increases in beta diversity, which I guess you would call heterogenization. I think that's kind of revealing that we have a word for homogenization, and we didn't really have a word for increase in beta diversity. Even though Sokolar has a nice paper showing that humans do lots of things that increase beta diversity, right? If I put a national park and then I have a big buildup outside of hotels, I've actually just increased beta diversity, right? So I don't think it's so shocking that humans can be increasing beta diversity. One more paper, and then I'm going to try and pull it back to some bigger picture. It just came out with Ines Martins, looking at change in body size. So this is not one individual, but this is average community body size. You take all the individuals in a community, you take their average body size, what's happening? First, I just want to note you could change two ways. You could have each species get a little bit smaller, or you could have a change in composition where you have more of the small species and fewer of the big species. Those would both result in a reduction in average body size. What do we find? The fish are clearly shrinking. The average body size of fish is smaller than it was 50 years ago, 30 years ago. Everything else, plants, mammals, we don't find strong trends. You can find cases where average body size is up or down, but there's not really strong trends. So fish are an interesting exception here. The other thing we found is the change is mostly due to composition and not to evolution within a species. And probably the most shocking thing we found is that even though we did find these changes in the fish, where the average body size went down, the total biomass, right, the sum of the body sizes was constant, right, which kind of goes with the fact it's a compositional change. But there actually seems to be this pretty strong regulation on total biomass in a community. If humans come in with saws and cut down the trees, there's not. But if you leave them even a little bit alone, the total biomass in a community is fairly constant. So, sorry, I lied. One more paper. This deals with the Living Planet Index. I know you have some folks here that have a really nice paper on the Living Planet Index too, which I think is very compatible with the result here. But in this one with Brian Leung and some others, we argued that, well, when you see a decline or an increase, it could be because there's this nice tight cluster of every system declining a little bit, or it could be because most systems are having no change and you got a few systems with drastic declines, which would still give you an average decrease. And of course, you could have the same thing in the other direction. You could be most systems not changing and a few extreme increases. And when people hear decrease, they usually think of this picture, but what we found the Living Planet Index indicated, and we actually did have a scenario where you could have both a cluster of extreme declines and a cluster of extreme increases, which would be a sixth scenario, but we didn't find that. What we found was this. The vast majority of species are showing no change, and there is a small fraction, about 3%, that are showing drastic declines in population size, which of course averages out to an average of a decline. But it's a pretty different story to have this story going on in your brain versus having this story going on in your brain. Okay, so all of which, especially those last two, well, so let me try and summarize this, and then I want to do some newer work on big picture. So it seems like what we're finding is something like a central limit theorem of increases, decreases, you know, no net trend in most of them, not all of them. There were some systematic changes. And what we found is in the terrestrial world, about half the land has been taken over for parking lots and agriculture. But the other half, the real story is not decline, but churn, turnover, change, right? Richness is not necessarily dropping. Abundance in this half is not dropping. What's really happening is the species composition is mixing up very, very, very quickly, seemingly quicker than pre-human times, although that's very hard to measure. The marine world is more, you know, there's more diffusion, there's more dispersal, and so you don't have this nice half chopped up by humans, half sort of left alone by humans, but you have this churn. The churn is actually stronger in the marine realm than it is in the terrestrial realm. Okay, so a couple of the results I've shown really suggests there's this strong regulatory central tendency of community properties, whether it's a total abundance, total biomass, total species richness. And indeed, we found exactly that result. There's this nice statistical test called the Augmented Dickey-Fuller test, ADF test, which can test whether something has regulated to have a central tendency or if it's a random walk. And it basically gives you a score. 0 is we don't know. Positive numbers is it's a random walk. Negative numbers is it has some sort of a central tendency to come back to the same point. It's not showing, in other words, it's not showing as much variance of divergence as random walks. So this is species richness. And you can see almost all of species richness shows at least a trend towards being regulated. Not all of them are statistically significant. The dark bars are statistically significant. Total community abundance, same thing, very strong. Almost all communities are trending towards a central tendency or regulation of species richness. right? And that you kind of came out of this slide, kind of came out of this slide, right? So this is really our attempt to start going back, and a lot of these results, some of these results didn't surprise us, a lot of these results surprised us. One of the things that surprised us the most is that it kept being centered around 0, right? I don't have the strong agenda some people do that everything humans do is terrible. Didn't expect it to be always negative, but I didn't expect to find such a strong clustering around 0. And that actually demands an explanation. And so I don't have time to get into deep speculation, maybe a conversation, we can talk about it, or you can read the paper. But what could be doing regulation on species richness or total community biomass? But it sure seems like there's some force that's doing that. Okay, I told you to take a look at the graph of change in population size, which comes back right here. It's the same as this graph. If you were like me, you looked at that graph, you said, it looks pretty normal. It's maybe the peak is a little sharp, but that's probably because the bars are weird. And that's what my research group thought for a long time. I think that's what most people think. But if you actually fit normal curves, they're not that great a fit. It's much more leptokurtic than a normal curve is, right? So this is a trend in species richness across this same data set, biotime. This is trend in Shannon diversity. This is trend in Simpson diversity, right? Hill number 2, hill number 1, hill number 0. This is trend in population size of the total community. I'm sorry, and this is trend in population size of individual populations. This is the trend in Jacquard similarity. This is a different data set. This is a change in phenotype. These are selection coefficients from Joel Kingsolver. This is the same data from a different data set. So this is BBS, this is the population growth rate, and this is the slope in species richness. And none of them fit the normal curve. They all have an excess of things close to 0, and they all have a paucity of things in intermediate change. And it's hard to see, but that ends up meaning they have an excess in the tails as well. So what we found is a deep dive into probability distributions. And it turns out there's actually a lot of logic, and statisticians probably already knew this, but most scientists didn't know this. There's a distribution known as the Subbotin distribution, named after a Russian probability theorist. And it's a generalization of the normal distribution. So this is a normalization constant, and this is the shape, right? If this was if alpha was 2, x minus mu squared, that would be the normal distribution. But if you vary alpha, you get different shaped curves. So if I take alpha equals, for example, 1, the dashed red line, I get this more leptokurtic curve. And if I take alpha equals 10, I get this more platykurtic, almost uniform distribution. Okay, and the one in particular I want to call out is the Laplace distribution with alpha equals 1. It has this very leptokurtic sharp curve compared to the normal distribution, which is the dashed black line. And if you plot it on a log frequency scale, the normal distribution becomes a parabola, which makes sense because it's x minus mu squared. And the Laplace distribution becomes this tent or triangle shape, which makes sense because it's the absolute value to the 1 power. And these are the fits of those data sets to the Laplace distribution with Monte Carlo error bars. Biodiversity change is not normal, it's Laplacian, right? This really shocked us when we discovered this. We started out with this conversation about black swans and fat tails and rare events, and we did find fat tails, but what we really found is a Laplacian distribution of trends in biodiversity. And so what does that mean? Again, the Laplace is also known as the double exponential, right? It looks like this if you plot it on a regular scale. It's just an exponential back to back to each other. And with the Laplace doesn't have to be centered on 0. We've ended up, I didn't belabor that point, but almost all of these distributions are very closely centered on 0, right? Which is and that's what I was just talking about, is the regulation. But the shape of the variation, for a long time we've said, gosh, there's a lot of variation. What is up with the variation? And I think it's a really important point. The Laplace says there's more stasis, near zero change, right? Trend lines near zero, and there's more extreme change, things way out in the tails. Which if you think about it is exactly the result we found with the LPI. We didn't find a clump way out on the right tail, but we found a big clump of extreme population declines, and then a curve that was exactly centered on zero with just a little bit of variation around zero, and that's exactly what a Laplace distribution predicts. I will spare you the deep dive that I kind of had fun doing into statistics and probability theory, but there are a number of explanations behind the Laplace distribution. When you have more heterogeneity in variance, right? When the variance is, for example, variance is different across species, you can very quickly get a Laplace distribution out. When you have these positive feedback loops where the rich get richer, you can quickly get to a Laplace distribution. The one that I find most fascinating, so you may know, right, so you have a random walk, and then there's an Ornstein-Uhlenbeck process, which is the typical physical model of a central tendency where there's a push back to the center. And the strength of the push back is it's basically the distance you've moved from the center. So if you're really far away, you get pushed back really hard. And if you're really close, you don't get pushed back very hard. But if you go to, and it turns out that if you do the same thing that you did with the Sabaton, so instead of taking the absolute value, you raise the difference that you've gone from the center to a power, it produces a Sabaton distribution with the same power, right? So random walks end up producing, yeah, something didn't quite work there, right? But random walks end up producing a normal distribution. And actually, if you assume that the pushback to the center is constant, so it's the same whether I've just deviated a little bit or a long ways, you get a Laplacian distribution. So here's the central regulation again. So I think there's some really interesting mathematical implications of the Laplace distribution, but I also think there's some really profound conservation implications. Don't pay attention to mean trends because most of what's happening in nature is flat, but do pay a lot of attention to the extreme changes, right? That's kind of, I know some people in conservation, especially species by species conservation, that's what we're doing. But I think a lot of biodiversity scientists and global conservationists have really gotten focused on mean trends. And this is really saying, actually, we need to go figure out what's causing the extreme declines and pay attention to those. Okay, last section, and it's pretty short. So just very quickly, and you're gonna have to participate by raising your hand in a poll here in a minute. 3 main reasons to preserve biodiversity. Ecosystem function, it helps humans, right? Flood control provides fiber in the source of wood, forms of wood or paper pulp or whatever. Pollination services to our crops. Biodiversity ecosystem function, it's more indirect. We actually need biodiversity to get that ecosystem function. The problem with ecosystem function, I'm steering the poll, but I'm going to go ahead and say it anyway. Problem with ecosystem function is a plantation of Pinus radiata or a plantation of palms can have very high ecosystem function, right? But it's a single species. So this is kind of trying to put biodiversity back in. Or a moral argument. Can I look my grandkids or great-grandkids in the eye while, you know, I was a voter while 50 species went extinct, right? Do we have a more, or even not my grandkids, outside of humans, do humans have a moral obligation to nature? All right, so real quick hand show. How many people think ecosystem, the question is, what's the best argument to make to society to get society to preserve biodiversity? First one's clearly an economic argument. How many people think that's the best argument? Few people. I know people don't like to raise their hands, but go ahead and raise your hand. I'm not looking, I don't know most of you. Biodiversity ecosystem function. We need diversity because it makes ecosystems healthier, basically. Okay, more hands, moral argument. The last two are about equal. Okay, I personally fall in the third camp, but I actually think this is an important empirical question. We're not necessarily having that conversation enough. Same thing, which emotion, if I want to get people to pay attention to biodiversity, I have parallels here with cigarettes and smoking. We certainly do push the cuddly button a lot in nature, right? I mean, who wouldn't want to conserve a panda, right? And this is not stopping smoking, this is keep smoking, this is selling cigarettes, but we push the, you know, cool, sexy, suave image quite a bit. There's the fear notion. Biodiversity is devastated. 70% of the planet's gone. It won't support human life. We're approaching tipping points. Scientists play this game quite a bit. Of course, it's been around in cigarettes since the 1970s. Here's your lungs on cigarettes. Here's your lungs without cigarettes. And here's a person who had to have an autopsy because they died of cigarettes, right? Social approval. Here's Smokey the Bear with a nice Boy Scout and a Girl Scout saying, you can help prevent forest fires. Turns out we prevented too many forest fires in the United States, but that's another conversation. And social pressure has been increasingly used in anti-cigarette campaigns as well. Or the hope message. California condor came back from extinction. You can do it. You actually can beat cigarettes. People quit cigarettes every day successfully. So same survey. These four emotions, which emotion do you think is the best emotion to push? If you're an ad, you've got $1 billion ad campaign, which emotion are you going to push in your ad campaign to get people to actually change their behavior for biodiversity? Is it the cuddly, cool? Okay. How many people think it's the fear? I'll just comment, this is the one that the scientists who are out there speaking to the press for us in most cases, maybe not in this room, but in most cases, scientists out there speaking to the press are pushing that button, and the press loves that button too. Social approval. Do it for your kids, do it for your neighbor. And how about the hope button? That's the most popular one. Yeah, so I actually, just two months ago, I was in Berlin. It was a science communication conference, and I gave a presentation, talked about the insect apocalypse and how it got covered in the press and what scientists thought about how it got covered in the press. But, and I presented this slide, and somebody else, who was a social scientist, actually got up and said, actually, we know this really well. Fear is a negative emotion. Negative emotions paralyze people and cause no change, except it makes people click and buy newspapers, right? But it actually, this is why politicians use fear, is it makes the other side stay home, right? And these are positive emotions, and these actually cause change. And it's not a coincidence the cigarette campaigns have taken, this was 1970s, this was 1990s, and this is today. So advertisers have known this for a long time, but scientists keep pushing the fear button. Okay, so let me stop, and then I understand we'll have a robust conversation. So wrap up, biodiversity is complex and high dimensional. It is not a single number. It is not easy to operationalize. It is clearly entrenched in policy. We couldn't get rid of it if we want to, and I don't want to, but we need to recognize it's not a simple thing to measure. My best answer, after working on this for 10 years, is rarefaction curves capture quite a bit. They're pretty simple to work with, and they actually have a lot of benefits, including incorporating abundance and scale and causes of change in richness. Local communities are very close to being random samples of regional pools. I'm not saying ecology is random, I'm saying we need to change the scale that we emphasize. We study biodiversity for both applied and basic conservation. Human impacts are mostly through high-intensity land use, straight removal of individuals. Outside of the biggest impacts are really turnover, right? We create winners who increase and losers who decrease. So we're changing the composition. It's not that we're collapsing biodiversity in most of these medium and low intensity places. Out of studying all of these trends, two surprising results. Biodiversity trends at a community level or even a population level seem to have surprisingly strong regulation. There seem to be forces causing central tendencies on species richness, total abundance, biomass. And it shows a little PLOS variation, which is very important because it's, to put it in a binary, it means no change or huge change. That's what Laplace is implying. And finally, I think ecologists need to get better at discussing biodiversity. Well, I think I flew by a slide there, but to just summarize it, I think we need to be smarter about learning from our social scientist peers, what they know about how to communicate and how to motivate behavior. And I also think, going back to the very first point of how do we define biodiversity, I think we have to start talking to people about how they define biodiversity, right? So we're very quick to say, oh, invasive species, those don't count, which maybe that's a good answer, but it's a normative answer. It's a values-based answer, and society is making values-based answers about biodiversity. I think we need to start incorporating the society's value-based answers about biodiversity. So I'll stop there.